Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the INSERM Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Almut Heinken, who's a junior professor at the INSERM Institute of Nutrition, Genetics, Environment, and Risk Exposure at the Université de Lorraine in France. And we're going to talk today about some really interesting work that she's been doing at the intersection of the microbiome, human genetics, drug discovery and pharmacogenomics, maybe more so, and actually how our microbiome influences the way that we process medicines and could be, I think, a big part of the future of personalized medicine. So thank you so much for taking the time today and looking forward to the discussion.
Yeah, thank you so much for the invitation. So yeah, I look forward to discussing my work with you.
Super. You've published a couple papers in the last year or so that really caught my eye. The first one was called, and I'll put links to these in the show notes, but the first one was called Genome-Scale Metabolic Reconstruction of 7,302 Human Microorganisms for Personalized Medicine. And the second one is called Apollo, which is on bioRxiv, a genome-scale metabolic reconstruction resource of 247,092 diverse human microbes spanning multiple continents, age groups, and body sites. And I just thought I'd mention those so people can go and have a more detailed read, because we're going to spend some time talking about that work and other things that you're focused on. I wonder if you could give a quick summary of the main things that you're focused on and interested in, and then talk a little bit about those two papers and what they focus on, and then we can take it from there.
Yes, of course. So yeah, in my work, I focused on systems biology modeling of host-microbiome interactions. And for that, I use a systems biology method called constraint-based reconstruction and analysis, or COBRA. And the COBRA approach relies on so-called genome-scale constructions. So these are mostly metabolic networks of a target organism. And so containing the reactions, metabolites, and the genes that encode the reactions. So in molecular detail. So they are built from the genome sequence, so the annotated genome of a target organism. And then, so these genome-scale constructions are extensively manually curated against information from the literature. So such as carbon source utilization, so fermentation pathways, so respiration pathways, and growth requirements of the organism. So like the kind of data you would find in biolog data sets. So basically the metabolic and biochemical traits of the organism. So these kind of data are then used to extensively curate these reconstructions, so to ensure that they can really predict the metabolic properties of the organism. And then as a result, these genome-scale constructions represent the knowledge base of the organism. So now this curation is quite time-consuming, so it can take 6 months or more if done fully manually. And now, of course, this is not possible when you want to reconstruct the human microbiome, which consists of thousands of strains. So already a few years ago, we started developing methods to make this curation process more efficient. So it was already possible to build fully automated genome-scale constructions. However, then these reconstructions need further curation. So these automated reconstructions to be able to actually be predictive and capture the traits of the organism, especially species-specific pathways. So all the way back in 2016-17, we developed a semi-automated genome-scale reconstruction pipeline. So it starts from an automated draft reconstruction. It then curates many draft reconstructions simultaneously against experimental data, so from the literature and from biochemical textbooks that had been manually collected. And these curation efforts led to the first resource of genome-scale reconstructions of human gut microbes, AGORA, so which at the time covered 773 human gut microbes. So each of these AGORA reconstructions captured the metabolic network of one human gut microbial strain. So enzymatic and transport reactions, around 1,000 or so on average per organism. And what we could now do with these AGORA reconstructions is to now build community models of the human microbiome or the gut microbiome. So linked together many of these individual strain reconstructions into one shared environment, so which then simulates the intestine. So in a way that they can exchange metabolites with each other. And then produce a total metabolic output, which roughly corresponds to a computation prediction of the fecal metabolome. And so these microbiome community models can then be personalized with the data of an individual. So the microbiomes of, for example, patients and controls. So the way this is done is that we retrieve the relative abundances of taxa from metagenomic sequencing data, so 16S RNA or shotgun sequencing data, and then map them onto the namespace of the AGORA reconstructions, and then link together all of the AGORA reconstructions that correspond to the taxa in one individual. And then to ensure that we also capture the ratio that these organisms were present at in the original sample, we then build a community biomass function. So we ensure that the microbes are growing in a ratio, so in silico growth predictions according to the ratio that was observed, so in the sequencing data.
That's super interesting. So if I replay that back to make sure I'm getting it right, step one is to build this kind of master model of the I guess all potential microbiome communities, or at least some estimate of the assembly of gut organisms that could influence the way drugs are processed as we take them, and then you can apply that model to take, I guess, a sample from any individual, and then use the model to make a more personalized version of that allows you to then simulate the impact of particular drugs that might go through their microbiome. Where have I got that right, and where have I got that wrong?
Yes. So basically, that was then later become possible with AGORA 2. So with AGORA, or with the first version, we basically capture metabolism on the genome scale. So not just drug metabolism, but also a variety of different pathways. So glycolysis, fermentation pathways, amino acid metabolism, and so on. And then, for each organism, we can then simulate growth. And then, so by linking together all the organisms in one particular sample, we can then simulate community growth. And we can predict what the community as a total is producing. So the metabolic output of one patient's microbiome, for example, compared with another patients.
And then once you know that, once you're able to make personalized predictions of the output of the gut microbiome, which diseases does that matter for? What do we know about the impact that has on? I know you worked in colorectal cancer, for example. Maybe that's a good example to start with. What do the differences between these individual microbiome communities drive in that disease, as an example?
Yes, so it has been shown for a variety of multifactorial diseases that the composition of the gut microbiome is distinct, or differs between patients and controls. However, now we don't. I mean, now what we're really interested in is how this affects the functionality, so the activity of the gut microbiome. And now you could also. You could get that through metagenomic analysis, but now gut microbes exist in a community, so there are species boundaries between them, so they can exchange certain metabolites with each other. So through this metabolic modeling approach, we can now predict metabolites, so the quantitative potential to produce metabolites by the individual microbes and the total community. And so I've applied that, and also other people have used the AGORA models for these kinds of simulations. So one type of complex multifactorial disease that has been clearly linked to the microbiome is inflammatory bowel disease. So there, it has been shown that the gut microbiome and the diseases that are taken together as inflammatory bowel diseases, so Crohn's disease and ulcerative colitis, have a very altered gut microbiome composition compared with healthy controls, in other words, they have so-called dysbiosis. And we were now interested if we could predict, using the AGORA models, how the metabolic capabilities of the gut microbiome differed in inflammatory bowel diseases. So in two papers I published back in 2019 and 2021, so we retrieved metagenomic samples from pediatric Crohn's disease patients and healthy controls. And we then built a community model from AGORA for each patient, each control. And then we asked the question, what could these microbiome communities of each individual theoretically produce? So the whole range of metabolites they could secrete so into the simulated fecal environment. And in the first article, we focused especially on bile acid metabolism. So it's well known that the human gut microbiome modifies human primary bile acids, so that were produced in the liver, and transforms them into secondary bile acids. And it has been shown in metabolomic measurements of feces that Crohn's disease patients have altered levels of secondary bile acids. And indeed, through the computational modeling, we could also predict that the bile acid metabolism of the human gut microbiome was distinct in the Crohn's disease patients, so compared with healthy controls. And not just the overall levels of produced secondary bile acids were distinct, but also the microbes that were involved in these transformations. So the Crohn's disease patients had increased involvement of proteobacteria such as Escherichia coli in bile acid metabolism.
And so it sounds like the models can tell you both something kind of fundamental about the disease biology, right? How the microbiome, what kind of metabolites it produces, and it can tell you about the altered state, but it can also be used to predict how certain treatments might be processed, right? I'd love to hear a little bit about that second piece. What diseases does our microbiome get involved in processing the treatments we take? I assume it's almost all of them, right? Because every small molecule that we take, every little white pill has to go in through our intestines and gets it doesn't get to the right organs just magically. It gets processed by our stomachs and microorganisms in there. And I hadn't really fully thought about the impact that those communities might have until I started to read some of this work. So maybe you could talk a little bit about that. How much is a medicine changed as it goes through your gut microbiome? And are we underappreciating maybe some of the impacts that has on the drugs that we take and the dosage and the impacts that it has? And what diseases does that apply to? Obviously, IBD you mentioned is a big one, but it sounds like maybe almost every disease this could be the case for.
Yes, so yeah, already a few years back, studies have shown that the gut microbiome is extensively involved in the metabolism of prescription drugs. So around 2/3 of prescription drugs have been shown to be chemically modified by the gut microbiome, and these influences on drug metabolism can be both direct and indirect. So in some cases, drugs that are taken orally, are directly modified by microbial enzymes. So in some cases, this leads to the drug being inactivated, so the bioavailability is reduced. It gets inactivated in the intestine before it can reach its target, such as the liver, for example. So in other cases, a detoxified drug, so a drug that had been inactivated and detoxified in the liver, gets converted back into its toxic form. So this can then lead to side effects, so increased toxicity of the drug. And in yet other cases, drugs are actually given as a prodrug. So the drug is given in an inactive form, and then microbial enzymes cleave the drug and release the active drug. So in these cases, drug metabolism by the gut microbiome is actually helpful. And then there are also indirect effects. So some microbial enzymes produce metabolites that compete with drug metabolites over human enzymes in the liver. So then this can then also have an effect on the human enzymes' ability to detoxify this drug and excrete. It's
super interesting. And I guess you've recently expanded this work through the second paper that I mentioned, Apollo, which looks not just at the gut microbiome, but 5 different body sites, also people from 34 different countries, a wide range of age groups. What did you learn from that on the differences between the output of these communities of microbes across countries, body sites? For example, where do we see the biggest difference? I would assume that guts and another body site are more different in a person than 2 guts between 2 different countries, but I'm interested in how those communities differ. And maybe you could talk a little bit about the different body sites that you looked at, as not every part of the body has microbial communities either, right? There are some parts that are sterile.
Yes, Apollo was built in a somewhat different way than AGORA 2. So for AGORA 2, we used reference genomes as the starting point. So genome sequences that had been applied from cultivating the bacteria. Well, Apollo relied on a cultivation-independent approach. So Apollo was built through curation of draft reconstructions that were built from metagenome assembled genomes. And metagenome assembled genomes are genome sequences that are retrieved directly from metagenomic samples. And this had been done through previously published studies. So by Parsoli et al. And Almeida et al. And these studies yielded around 150,000 and 90,000 metagenome assembled genomes, respectively, from around 9,000 or so metagenomic samples from all around the world. So these metagenome assembled genomes, or MAGs, covered a wide variety of microbiome samples. So from different body sites, so from the gut, the skin, the vagina, and the oral microbiome, and also from all continents. So from Europe, the United States, Asia, so China and Japan, but also non-Westernized communities, so including from Africa and South America. So as a result, these resources of MAGs greatly increase the diversity of available genome sequences for the human microbiome. And we now use these two resources of MAGs to build a genome-scale reconstruction for each MAG. So then resulting in around 250,000 or so genome-scale reconstructions in total. And from this large resource, which is called Apollo, we could then build around 14,000 microbiome community models. So that accounts for a wide variety of disease states. So we have multiple body sites. So we have just gut microbiomes, but also skin and vagina, as well as the nasal microbiome. Then we have also all age groups. So in particular, this allowed us to compare adult and infant gut microbiomes. We can have patients with inflammatory bowel disease. With Parkinson's disease patients. And we have gut microbiome samples from patients with infection. So which we again could compare from controls, from healthy to healthy controls. And then another comparison we could perform is preterm infants versus infants that were born at term. So we could, for all of these microbiome community models we had built, we could then predict the metabolic potential and the pathways present in each microbiome, and then analyze what was different between for example preterm and term infant gut microbiomes. So we could also perform so we could also apply a machine learning algorithm, random forest, to the reaction content and in these microbiome communities. And we could see that they were clearly distinct, and we could retrieve which reactions really could really distinguish for example preterm and term infants, or healthy microbiomes in the gut, in the skin, and in the vagina.
If we dive into the term birth versus the preterm birth, just to give people a sense of what those differences are like I think you've got a complex community of microbes, and I guess differences across all of those different body sites that you mentioned. What does it look like when you say there's a difference between term and preterm? Is this the types of microbes that are present, their abundance, the output of metabolites that they have? Maybe you can just give a flavor of when you look and you see a difference. Is it one that would be obvious to someone in the sense that you can describe the communities to say there's much less of this organism and there's much more of another? Or is it more subtle where you need advanced algorithms or something to detect the differences, and it's not a, kind of simple to explain change in the ecosystem.
Yeah, with these microbiome community models, we are not just analyzing which taxa are increased or depleted in the different conditions, but we can also predict which pathways are higher or lower in abundance, as well as how this should translate into function. So the metabolite levels that can be produced. And so how distinct this is between so different disease groups and healthy controls really depends. So for example, for preterm and term births, we see a very clear distinction. So when we cluster these microbiome community models by their reaction content, we see very distinct clusters.
Interesting.
So we also see distinct clusters, for example, between gut microbes of healthy and undernourished infants, and of healthy adults with and without infection. Well, for other comparisons, for example, so obese gut microbes versus normal weight individuals, there we see far less difference. So there the stratification of the different groups based on their metabolic pathways in their microbes is less clear.
Yeah, interesting. And what about the impact of diet? You talked a little bit about there's a clear difference between undernourished and nourished. What do we know about? I suppose there's some circularity that we have to think about as well, that your diet influences your microbiome, and then your microbiome in turn influences your nervous system and the way you make decisions and satiety signals. What do we know about the interplay between diet and microbiome based on either the work you've done or what you know more broadly about the field?
So clearly the diet has a large impact on the gut microbiome. So we know that the composition of the gut microbiome depends so varies depending on lifestyle and region, so between countries, and the dietary habits in different cultures are also different. As well as so the gut microbiome also differs, the gut microbiome composition also differs, for example, between vegetarians, Western diets and more healthy diets, so high in fiber. And one advantage of these microbiome community models is that we can contextualize them with a personalized diet. So we have built a website called the Virtual Metabolic Human. So that holds a database of around 1,000 or so different food items. So if you now know what you ate in a day, so the quantities of different foods such as bread or pasta, you can enter that into the database and then design simulation parameters that can then be fed into the community models, and then predict what the microbiome is producing on this particular diet. So if you have, for example, food frequency questionnaires for the study participants, so that the metagenomic samples were from, then we can also contextualize each microbiome community model with a personalized diet.
Interesting. So you can you basically provide the diet and other factors like disease and other sorts of things. I'm just checking out the website right now as inputs, and then you run the model and can then effectively simulate or predict the metabolic output. Is that right?
Yes. Now, for example, now you could also predict how different diets what influence the metabolic activity of the microbiome. Also, you could predict targeted diet interventions, such as prebiotics.
Wow.
So metabolite or like fibers that can serve as a carbon source for beneficial microbes. So another resource we have developed is the whole body model of human metabolism, which was built from a global reconstruction of human metabolism, and which now integrates all of the organs in the human body into an anatomically correct whole body model of human metabolism. So this whole body model is sex specific, and it can be contextualized with physiological parameters such as weight or height, and also with a personalized diet. And it can be then integrated with the community model of the microbiome.
That's fascinating.
So that way you can then predict host, diet, gut microbiome interactions. So the effect of diet on both the host and the microbiome, and also how the interplay between diet and the gut microbiome affects far away organs such as the brain or the heart. And there then you could also integrate the drug metabolism. So you can also predict what would happen if you gave this simulated human a certain drug. So the level of gut microbiome activity that would transform or degrade the drug, and how much of the drug then would actually reach the liver.
Interesting. And how accurate are these predictions? If you're able to measure some of these outputs on the other side, obviously we know and don't expect that it's going to be perfect, but I'm curious how accurate they are. And once we have an understanding of that, where are the gaps? Like, which parts of it are highly accurate? I would assume maybe the impact of diet on microbiome is highly accurate, but or more accurate, but some other things that you predict maybe less so because you have less data. Maybe you could talk a little bit about that, where the gap is between if you're able to get an empirical measurement versus the prediction, and what drives the difference.
So yes, of course, the models, well, they are computational models. So in their basic form, they are an oversimplification of a real organism. So one disadvantage is that they, in their basic form, don't account for regulation. So regulation of metabolism or kinetic parameters, which on the other hand is also an advantage, since then you don't need these parameters to perform the simulations. And yes, so now for drugs, that has the disadvantage that currently we do not capture pharmacokinetics, but in the future, these genome-scale models could also be integrated with pharmacokinetic modeling. So then that way we could also predict the dynamics of drug clearance and absorption in the human liver. And another thing is that since these genome-scale constructions are knowledge bases, there will always be gaps in the available knowledge that goes into these reconstructions. So if gene functions have not been annotated yet, then we cannot include them. Or if pathways just haven't been discovered yet, then it's also currently not possible to include them. And the predictive quality depends on the level of curation that was performed. And for these AGORA 2 reconstructions, so we have over 7,000 reconstructions, and we performed a lot of curation, but there was just a limit to how much we could do. So we still see, though, that if we now compare the predictions of AGORA 2 against independent experimental data, so we get a very good accuracy of 0.8 or so. So so the predictions are quite good, but we could always do more creation. And now for drug metabolism in particular, there are a lot of microbial drug metabolism just probably still needs to be discovered. So in order to include these drug metabolism by the microbiome, so we need to know either the microbiome enzyme that performs these transformations or the microbes, so the species. And if we don't know that, then we cannot include it. And we also need to know the stoichiometry of the transforming reaction, as well as the structures of the drugs. So in future expansions, we could further and further expand and refine these drug metabolism pathways. So to account for new knowledge as it becomes available.
Yeah, super interesting. It's a good maybe segue to the future. We've only got a few minutes left here, and I was interested in closing out on what's next for you. You've done a lot of really great work. I think you've had a very productive last couple of years, but you're probably thinking about what's next and working on a few other projects. So what else are you working on right now? What are you most interested in as a follow-up to all this?
So currently I'm particularly interested in the infant gut microbiome.
Yeah.
So in our institute, we are very interested in epigenetics and in early life influences and exposures that could predispose infants towards metabolic diseases such as obesity or metabolic syndrome later in life, so which is referred to as fetal programming. So the viability of B vitamins such as folate and vitamin B12 also plays an important role here. So in the work I'm currently doing, we modeled infant gut microbiome. So we retrieved a cohort of 20 infants, around half of which had been delivered vaginally and half to caesarean section. And we had metagenomic samples at 4 time points during the first year of life. So we built a microbiome community model for each infant in each time point. And then we asked the question first, which metabolic pathways were different between the infants born to caesarean section and the vaginally delivered infants. And found that especially at very early on, so at 5 days or so and 1 month, the infants delivered to caesarean section, or rather their gut microbiomes, were depleted in metabolic functions. So in particular, they had a reduced capacity to utilize the so-called human milk oligosaccharides, which are found in breast milk. And also a reduced capacity to synthesize a variety of bioactive metabolites, such as, for example, branched-chain fatty acids or pyruvate, and also GABA, so a neurotransmitter. And moreover, we compared the metabolic capabilities of these infant gut microbiomes with adult gut microbiomes. And there we also found that the two were clearly distinct. So certain fermentation products, such as propionate, were clearly enriched in the adults, while others such as lactate were enriched in the infants. And also the infant gut microbiomes had a much higher capacity to synthesize B vitamins, such as folate, or riboflavin. And this has now led us to the hypothesis, so based on these computation models, that the infant gut microbiome could provide the host, so the developing infants with B vitamins, so which then could also affect its development.
Interesting. How much does this change into year 1, year 2, year 3? Is there a point at which the gap and the differences close, or do you see that gap persist into childhood? What do we know about that?
So from what we see is that the metabolic functions of the gut microbiomes became comparable from around 6 months of life on. So the effect of caesarean section doesn't last that long, though it could still have an influence. So in particular, it could influence epigenetic regulation. So very early on.
Yeah.
So, and then which might then also leave an imprint. And then in future work studies, then we will also look at cohorts where we then have data from the infants later in life, or where we can compare, for example, breastfed and formula-fed infants.
That's super interesting. I'm just thinking about, we've interviewed a couple of newborn screening programs recently, and there's a whole lot of interesting collaboration and science to do around just looking at the variation in different types of factors in birth, everything you mentioned, different diseases, different kinds of diets and lifestyles, and just getting a better understanding of how do we set kids up for as much success as possible, because it's very much art rather than science right now in a lot of cases, isn't it?
Yes. Yeah, actually in our lab, we are also very interested in inborn errors of metabolism.
Yeah.
So we have a Center for Inborn Errors of Metabolism at the hospital. And I recently published a review article on the potential of constraint-based modeling to elucidate metabolic differences between patients and inborn errors of metabolism. So since it was generally assumed that the patients are all very similar because the diseases are generally based or are caused by a single mutation, but there we also see differences between patients. So some patients may have very mild symptoms, where others may be more severe. So there we think that these personalized genome-scale models could also help us understand better how these relatively simple diseases could serve very different presentations.
Yeah, that's so interesting. I'm just looking up the paper in the background right now. I'll just a quick read for people, and we'll put this link in the show notes as well. But it's called Towards Personalized Genome-Scale Modeling of Inborn Errors in Metabolism for Systems Medicine Applications. And you make a really good point here, which is one that I work with a lot in a lot of rare diseases, and it's a lot of the work that we do. And patients and families always make the point that while we're a rare disease, it's extremely heterogeneous. Every child or every adult with this particular rare disease often has very different symptoms. And if you assume that it's rare and it's therefore simple, you can miss a lot of the heterogeneity, which could drive treatment outcomes. It's going to change the prognosis for different patients. So I think it's really interesting. You're effectively building a gut microbiome model or a metabolic model for each of these cases that allows you to get a clearer picture behind what's normally just considered to be a monogenic disease that doesn't discriminate beyond the gene, right? But helps you get a clearer picture into what's driving those differences.
Yes, and here for inborn errors of metabolism, the gut microbiome also play a role, especially in these diseases where you have acids such as propionate accumulating. Where then the gut microbiome could negatively influence so these diseases.
Are you familiar with something called autobrewery syndrome? I learned about it relatively recently. Have you come across it?
Yes. So that's if the gut microbiome produces a lot of ethanol.
Yes. I think I'll check if this is apocryphal or not, but yeah, the gut microbiome produces alcohol basically, and it obviously results in liver damage over time if it's not addressed, because the body is essentially producing it can be reasonably high grades, but I believe historically there's at least one case where someone was pulled over for drunk driving, but it actually turned out they hadn't had anything to drink, and it was the issue with their gut microbiome that was throwing off the breathalyzer signal. And it just blew my mind to learn about this, to think about the ways in which our microbiome can be incredibly helpful, but can also cause real problems and unexpected ones, at least for me, and learning about some of these really extreme cases of dysfunction that don't come up very commonly, but obviously are a big issue when they do.
Yes.
Well, I'd just like to say thank you. This was a really interesting episode. I learned a lot. I think you're doing amazing work, and it sounds like there's a lot of hard work that goes on behind the scenes, assembling these incredibly complex models and data sets. And it's amazing that you've made it available to the community as well through the free online resource. I'll add a link to that as well. It's vmh.life, I think. I was looking it up in the background, but thank you. Thank you so much for taking the time for doing this.
Oh, thank you so much. And yeah, thank you for your interest in my work. And yes, so as you just said, so we have made all of these resources and the community modeling tools really available. And yeah, we have seen a lot of people in the research community use these models for very interesting applications. And yeah, we hope that AGORA 2 will also enable some interesting studies. Yeah, and I'm also available on, for example, the COBA Toolbox GitHub. Yes. And the COBA Toolbox Google group, for example, to answer questions. So about, the methods to apply, these, community models.
Super. Well, thank you. And thanks, as always, everyone, for listening to this episode. If you liked it, we'd really appreciate if you shared it with a friend or colleague who you think would enjoy it as well. And of course, leave us a review in your favorite podcast player. And as always, you can reach us anytime for feedback at podcast@sanogenetics.com. Thank you so much, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.